Hello everyone, and welcome to today's webinar hosted by the International Irradiation Association. My name is Arthur Dumba and I'm the General Manager of the IIA. I will be your host today. We're happy to have presenters and attendees from around the world joining us and appreciate your taking the time to be here. A particular welcome to IIA members joining us today. Your support makes events like this possible. All IIA activities, including this webinar, are funded through membership. Before we get started, a quick introduction to the IIA. The International Irradiation Association is a not-for-profit association that supports the irradiation industry in its entirety, encompassing all radiation technologies, applications, sectors, and stakeholder interests. We promote the safe and beneficial use of irradiation and act as the bridge between science and industry. The IIA works to advocate, communicate, and educate on behalf of all our members and the global community. Membership with the IIA offers numerous benefits, including access to presentations, publications, and resources, participation in our topical working groups, visibility and promotional opportunities, and networking and collaboration with fellow members and our industry partners. You can learn more about the IIA's work on our website, iiaglobal.com. There you'll find all our initiatives and resources, as well as details on how membership can support your organization. If you have any questions, please do not hesitate to contact us at info@iiaglobal.com. Today's webinar is titled Packaging Considerations for Radiation Processing. And we're joined by 3 expert presenters who will be sharing insight on key packaging considerations for radiation processing, including radiation packaging integrity, material behavior under irradiation, regulatory consideration, analytical testing strategies, virtual dose mapping, and modeling real-world conditions. I would like to start with a few housekeeping rules. This webinar is being recorded. A link to the recording will be shared with all participants shortly after the webinar is over. You can submit questions at any time using the Q&A function at the top of this screen. We'll have a dedicated session for questions and answers at the end of the session. We will endeavour to answer all your questions, but if we run out of time, we will circulate unanswered questions at a later date. The chat function has been turned off for this webinar, so please make sure to direct your questions to the Q&A tab. I would now like to introduce Wendy Mach from Canyon Labs to begin the presentation.
Hello and welcome. I'm Wendy Mach. I'm the Senior Director of Technical Services at Canyon Labs, and I have the privilege of kicking off this webinar by talking through the impacts of radiation sterilization on packaging systems. This topic often gets less attention than product sterility or bioburden control, but packaging can make or break a sterile barrier system. Whether you're validating a packaging change, developing a new system, or even auditing a supplier, it's important to understand that material performance under radiation is essential for product quality and regulatory compliance. So here's what I'm going to cover in the next few slides. Why packaging needs to be considered early and intentionally when radiation is part of your sterilization modality. Then I'm going to touch on underlying chemical mechanisms, what radiation actually will do to the polymers. And then we're going to touch on regulatory bodies like FDA and ISO expectations in terms of validations, and then common pitfalls like assuming cross-compatibility throughout sterilization methods or skipping long-term studies, which never happens. I'm also going to include a couple real-world case studies so that I can highlight packaging failures, how they occurred, and how they can be avoided. Again, as Arthur said, if you have any questions throughout this process, drop them in the Q&A and we'll tackle them at the end. So let's get started. Why does packaging matter in radiation processing? So radiation sterilization, whether we're talking about gamma, E-beam, or X-ray, is incredibly effective, but also indiscriminate. It doesn't just sterilize your product, it also interacts with your packaging. If your packaging materials and seals degrade, even subtly, they can actually compromise sterility, shelf life, and compliance. And in the end, this is why it matters. It's not a packaging problem per se. It becomes about patient safety and business risk. One specific failure that I encountered throughout my experience was when a medical device company used a gamma dose of 25 kilogray to sterilize a surgical tool. This is actually a very common dose. And what happened was the package passed the initial packaging validation. We did burst testing, dye, and some aging. But 6 months post-commercial distribution, what happened was they started receiving customer reports of seal failures. Some of these seals were actually peeling unevenly, while others actually tore the pouch. Investigations found that the polyethylene seal layer had actually embrittled due to the gamma exposure and the aging. Unfortunately, this resulted in a recall, a full packaging redesign, and unfortunately, $2 million in remediation costs. So the point of this study and story is to understand that early packaging evaluation isn't optional. It's a critical part of product safety and life cycle planning. Let's get into how radiation affects materials. At a molecular level, ionizing radiation, including gamma, E-beam, or X-ray, creates free radicals in polymers. These are reactive species that initiate either chain scission or cross-linking. So chain scission is considered to be breaking long polymer chains, reducing molecular weight, and then flexibility. It results in reduced elongation at break, lower tensile strength, and sometimes can increase haze. It's common in PP, PVC, and then EVA blends. Cross-linking forms bridges between chains. This often increases the stiffness and alters sealing properties. This can be found by increasing modulus and melting temperatures, which then affects seal behavior in our form-fill seal processes and is common in polyethylene and then certain nylons. These effects aren't always linear, though. A material may initially benefit from some cross-linking, but then once they pass a threshold, it becomes brittle or seal incompatible. This is particularly important when we're talking about multi-layer films. A PET outer layer may cross-link, whereas a polyethylene inner layer may then embrittle or yellow. So what does this mean for engineers? It's understanding that the entire laminate structure of the film or packaging materials must be evaluated. Don't just focus on a single material spec sheet. It also means that processing parameters like dwell time, dose uniformity, and cooling can actually influence that material response. Chain scission has a way of breaking things down literally. So if your packaging starts cracking under stress, it's probably just expressing some deep molecular trauma. Specifically, we're going to talk about degradation Oh, sorry. Next, I'm going to talk about degradation. When we're looking at this in practical terms, Tyvek 1073 is really well for gamma and E-beam. When we're talking about co-extruded polyethylene structures, you're going to start seeing yellowing, especially if you're going to look at doses above 35 kilogray. And then PVC and polycarbonates, you may have discoloration above 25 kilogray. Then you start bringing doses into this equation, and we're usually talking about a range, not a single value. So your validated dose range will usually look at 25 to 40 kilogray. And you have to ensure that your packaging can tolerate both ends. And when we're doing that, we're looking at what a max dose is that your packaging can withstand. Finally, when we're talking barrier properties, we need to understand how they diminish or maintain their stability over time or with storage. A packaging failure doesn't always occur immediately after irradiation. Over time, materials can degrade due to residual radicals and then also thermal aging. It's important to watch for a couple of tests that will help you evaluate this. And one of those is OTR, or oxygen transmission rate. If this increases by 15 to 30% over a 16 to 12 month post-irradiation process, you may want to start looking at a few things. WVTR also, especially if you're working with thin PE or metallized films. Additionally, delamination in adhesives or tie layers under heat or humidity cycling. So keep an eye out for those three things. And then, of course, when we're doing aging studies such as accelerated or real-time aging, these will also help you understand what those risks are associated with material degradation. Let's talk about regulatory expectations. What does a regulator expect from you as a medical device manufacturer? Or well, the first thing they're going to start looking at is whether you're fully compliant with 11607-1 and -2. And for those of you that aren't aware, we're currently in the process of creating a -3. In these standards, we're looking at understanding what aging, distribution, and seal testing is around packaging and how that is interacted with the product and sterilization. So making sure that you have evidence that your packaging withstands your worst case dose and your shelf life is where you're going to pick up your 11607 compliance. 11137 is your sterilization validation, setting your dose, and ensuring you're following the regulatory standards there. It is also important to make sure that you are taking care of your dose mapping, again, understanding your material compatibility. 11607 captures those real life aging studies and performance testing, and then including part of that, you're going to need to make sure that you have the data, which is presented using dye or bubble tests and peel strength. Additionally, you can take a look at microbial barrier testing, a comparison between before accelerated aging and then after accelerated aging to demonstrate that there's no degradation in those material barrier properties is an included data point. And then finally, a visual inspection criteria, making sure that as time evolves, we're not seeing yellowing or warping. It's important to recognize that validation isn't just about passing tests, it's about proving that the long-term packaging performance under real-world conditions is acceptable and documented. All right, let's see if I press the right button this time. Yay. Okay, common pitfalls to avoid. Now that we've covered science and regulatory framework, what are some of the missteps that teams usually make during a packaging validation for radiation checklist? So the first thing to do is to make sure that the material you're selecting is compatible. Many times people will pick a material that they're commonly associated with and an off-the-shelf package and not think about things like sterilization compatibility or design. Because the material can't withstand the dose you're using, it's always important to make sure that we're understanding the impact of the dose range, of the sterilization modality, and the package and product interaction. Always confirm compatibility through testing, not assumptions. Now, on that though, there's a fabulous document out there, TIR 17, that can help guide you through what some of those interactions are based on data that's out there, publications, etc. So if you're looking for a starting place, TIR 17 is a really good starting point. The second thing to think about is ignoring seal degradation post-irradiation. Even if your packaging seals are properly validated before sterilization, radiation can weaken or distort these seal layers, especially, like I mentioned, in multi-layer film. So in understanding seal integrity before and after dose exposure is critical. What is a statistical difference? And making sure that you set your acceptance criteria appropriately. Skipping comparative studies. If you switch from gamma to E-beam or potentially adjust dose ranges to widen the range, don't assume that your packaging will behave the same. Subtle changes in modalities could cause performance differences, and they should be validated or justified. Assuming that accelerated aging replaces real-time studies. So this is actually a very interesting one. I thought that we were moving away from this as an industry, but I continue to see that a lot of products are going to market without the real-time studies even being initiated. And the 1160 standard states that that needs to be initiated before commercially releasing your product. But accelerated aging is valuable. It is a worst case understanding of how your product will interact with sterilization over time. But the requirement is to do real-time data. And I have seen cases where the real-time data actually failed and the accelerated aging passed. Now, there were specific instances associated with that, but it is possible. Making sure that you understand that and when to schedule those as part of your validation is critical, because a lot of times this is where a lot of your timeline will get sucked up. Accelerated aging, even though it's accelerated, will continue to cover a lot of time. 1 year in a chamber is still 46 days. So making sure the packaging is brought in the very beginning is one of those critical points. Then neglecting transit simulation. This is an interesting one because a package, I've not seen a package where it's made from the manufacturer and used immediately. It does go through some type of transportation, and ensuring that that transportation simulation is accounted for in your validation is critical. Just because a package survives sterilization and storage, it doesn't mean that it will continue to survive the shipping. Vibration, drops, and climate exposure are all hazards that will expose weaknesses that weren't previously obvious in lab conditions. So each of these pitfalls are very avoidable, and with upfront planning, working with SMEs, and doing a comprehensive validation will minimize this. The earlier you test and stress your packaging system, the fewer surprises you'll be faced during your audits, your production, and fortunately, post-market surveillance. So one of the things that we're starting to see questions on are what considerations do I need to think about when I switch from gamma to X-ray? X-ray is becoming a new, it's not a new technology, but companies are investing in the equipment and the process. So there's a lot of questions around how do I switch between these two approaches? And it's not as simple as saying it's a dose to a dose, especially when it comes to packaging. You need to make sure that even if the dose remains the same, that you are considering carefully what that packaging change looks like. While both of these are ionizing radiation, they can differ significantly in how they interact with materials. I put up a couple of the differences between the two approaches here. Gamma radiation is emitted from a radioactive isotope. As we all know, it's typically cobalt-60 and will offer a deep, even penetration at a slow dose rate. When we're talking about X-ray, which is generated electrically, it also provides that deep penetration, but with a different energy distribution, and it can also have a higher instantaneous dose rate. So these physical differences can translate into distinct chemical and mechanical effects on packaging. For example, seal performance could shift. Some multi-layer sealants that tolerate gamma may soften, shrink, or degrade when we're looking at X-ray. Additionally, barrier films could exhibit accelerated aging due to the difference in sensitivity around dose rate or energy absorption patterns. And then there's always the potential for visual changes like yellowing or cloudiness. And that may appear or worsen more under X-ray if you've never seen it under gamma, depending on your materials. So what are the key things that I would consider before switching is making sure that I do comparative testing, looking at appearance, seal strength, or some chemical tests like OTR or WVTR. Look at existing aging data and determine if I need to redo any accelerated studies or real-time studies. Make sure that we capture all this documentation in our DHFs, design history files, and then making sure that we're notifying our regulatory bodies as applicable around any packaging behavior changes or shelf life claims that could be affected. In short, what I'm saying here is don't assume that a gamma to X-ray is a plug-and-play switch because they're similar modalities. Even minor changes in material performance can cascade into long-term sterility or compliance risks. So what are my key takeaways? As a wrap up, I have a couple core messages I want you to leave with. Plan packaging early in product development. If you've ever been at any of my webinars, I almost always end with that. Make sure you bring the right SMEs and the right engineers in the beginning to help make sure that your timelines are managed appropriately. Radiation impacts more than sterility. It transforms your packaging materials. Things like chain scission, cross-linking, seal degradation, and loss of barrier properties are real risks, and they happen all the time. Material selection matters. Not all polymers are created equal when it comes to surviving ionizing radiation. Make sure you're considering each layer, not just the outside layer. Dose range and modality must be validated. Don't assume a gamma equals E-beam equals X-ray. Performance will and can vary widely. Validation isn't a checkbox. I know there's a big sweep in industry right now to move away from checkbox approaches. It's an ongoing process, making sure that you're watching seal integrity, aging, barrier performance, and transit simulation is included in your validation protocols and executions. And then finally, getting ahead of problems through design and testing. The earlier you test, the more options you have, the fewer surprises you'll be faced with post-market. Ultimately, successful packaging validation ensures product sterility, regulatory compliance, and then patient safety. It's not just a technical requirement, it's a strategic advantage. It doesn't matter if you're using polyethylene, polypropylene, or polyester, picking the wrong material and you might go from poly to sorry. And that is all I have for this part. I'm going to hand it over to David. Thank you.
Thanks, Wendy. All right, can everyone see my screen? I hope so. Give me a thumbs up if you can, somebody. My name is David Gallegos. I am the Director of Analytical Laboratories here in Salt Lake City for Canyon Laboratories. And today I will be talking about packaging considerations for radiation processing from a chemical perspective, and really trying to highlight why this matters from a product safety, compliance, and performance perspective.
Sorry, David,
So,
would you be able to go to full screen mode, please?
Yeah, sorry about that.
Perfect.
All right, that's better. All right, so diving into kind of what Wendy talked about, how radiation can alter polymer structures. As she touched on, you can have both chain scission and cross-linking type effects after radiation treatment. And whether it's chain scission or cross-linking, both can be good or bad. In general, you don't really want scission to happen for compliance issues because as you can see, when you have a monomer or when you have a polymer that gets broken down into smaller molecular weight monomers, that increases the solubility, which means that you have a greater likelihood that those new monomers can migrate from your packaging into your product, be that a medical device or a pharmaceutical. In some cases, cross-linking can be desirable because it can increase rigidity and durability of your packaging system, but too much can lead to brittleness, discoloration, and undesirable effects. So these are things to consider when you're doing your sterilization validation, deciding on a dose. When you think about the dose, you want to sterilize and kill these microbes that's altering their DNA chemically. So that can also alter the chemical properties of your packaging material, right? So it's a balance of getting the right dose to where you're sterilizing, but also not completely degrading your packaging materials from a chemical perspective. So when you get polymer degradation and small molecule byproducts, there's also the possibility of potential toxicity of those new degraded polymers. So that's why we have to do chemical analysis as part of this process to make sure that our patients are safe and we're maintaining compliance. One test we can do to do that is extractables and leachables. And you want to perform this test post-irradiation due to the fact that you can form new extractables after the radiation process. And these molecules can then become leachables, meaning that they can migrate from your packaging into your drug product, or medical device. And you have to do testing both on the packaging materials or container closure, as well as the final drug product or medical device, just to determine compliance and everything is safe. So that involves identifying all these potential small molecules that you're generating, and then doing a TRA to make sure there's no toxicity associated with that before you can get approval for your final product. Some of the analytical approaches that we can use to assess changes in packaging post-radiation: LCMS, which is liquid chromatography mass spectrometry. This is an analysis for non-volatile chemicals. We also do GCMS, or gas chromatography mass spectrometry, for semi-volatile chemicals. And we want to make sure to do both tests just to cover the complete array of possible chemicals that are generated, so we don't miss anything. We can also perform FTIR, and this is a graphic that I show here. We use this to compare materials, sterilized and non-sterilized, so we can see any chemical changes that may pop up after sterilization, and you can compare the differences. It's an easy way to just visualize if there's new functional groups that have occurred after sterilization, any degradation, we'll see it here. So making sure we're compliant with different USP and ISO standards. These are some of the standards that outline the chemical testing that you need to perform to be compliant. So USP 661, we have both 0.1 and 0.2 now that will be official in December of this year. These describe basically the necessary, depending on the material that you're using for your package, you have to perform these tests to make sure that the material is well characterized, basically. So if any degradation had occurred, you would be out of spec on the different tests involved in USP 661 testing. So there's UV testing, TOC, FTIR, a few different tests that just make sure that basically your material hasn't degraded and it still fits with what it should be, even after radiation. Then you also have the USP 1663 and 64. These describe the extractable and leachable process. From a high-level overview, extractables is an exaggerated study that is kind of a worst-case scenario. You determine all possible chemical compounds that can come out of your packaging system. And from there, you would take that in the leachables test and see what actually did migrate from your packaging system into your final product. And then ISO 1137, Wendy talked about this, and this is just a whole suite of documents that make sure that your sterilization process is done properly. And all these things tied together, you do them, you'll ensure your compliance and safety of your patients. Now I'm going to talk about just briefly some real world examples of work we've done and what we've seen. So without getting too far into the weeds, I know this is pretty complicated. This was a pouching material. We did a water extract of this packaging material. It was a PET-based material, and it failed the USP 661 test for UV absorbance. So we needed to dive deeper and see what was causing this failure. And this is a LCMS test here showing UV at 254 nanometers. You can see there's a lot of peaks showing up, which makes sense because it failed the UV test in USP 661. And so our job now was to identify all these peaks and figure out where they're coming from. So this is why it's important to really get that sterilization validation right, because if the dosages are too high and you see all these fragments, it's just going to cost you extra time and money, which is good for me, but not good for you as the person who's trying to get this approved. So basically, we were able to, with mass spec, determine the masses of all these unknown compounds, and each peak here in this chromatogram is a unique compound. And we knew the material was PET, so we were able to match it with some standards. And we saw that, as I kind of described earlier, the polymers had broken into these smaller monomer compounds. These are just different like terephthalic acid moieties, so they're the smallest monomers of the bigger polymer. They broke up as a result of radiation, and now you have all these UV active compounds that are causing you to fail some of these analytical tests that you need to perform. So once we've identified the individual peaks, you can then do toxicity studies, make sure they're not going to cause any effects on patient safety. We can quantify the levels that they're present in this water extract. And basically, you have to do that for each peak and go from there. And if it's safe, then you'll be good to go. But hopefully you can avoid having to deal with all this if you can select the right dose in the first place. Here's just an example of the LC-MS data we get to determine these unknown compounds. So we're able to determine the mass. Knowing what the original material was, we can start to kind of piece some things together that make sense. And experimentally, if we hit this parent molecule with the mass of 306 with high energy, we can force it to break up into the smaller components. And these compounds generally aren't available to purchase as standards, so we can't officially quantify these compounds, but they're just putative structures based on the data we've collected. And you can see that back up here, we had at 307 and 265, we had these masses in here. So basically all these peaks are just larger and smaller monomers of the parent PET polymer. And that's kind of the process we go through in an extractable study to determine unknown compounds when we see those. So the key takeaways here is that you want to select the right material. You want to do your radiation processing correctly, ensuring the right dose to make sure that your packaging is going to perform from a material perspective. It doesn't break down, the sills don't degrade, you don't have degradation of polymers into monomers. And so you have to do chemical testing after radiation to ensure that you didn't have degradation chemically and that you're all compliant there. Then you would go to regulatory review and make sure from a toxicity standpoint that everything is good, and you can go to product release and approval. So Canyon Labs can help through this whole process. We offer USP 661 testing, extractable leachable testing, FTIR for material characterization, and consulting throughout the whole sterilization validation process. Thank you, and I will now pass it to Tobias.
Hi, everybody. I hope you can see my screen okay here. And I will change here a little bit to go into virtual dose mapping as a tool to inform how you design your packaging. And I think I give you a quick prime on radiation sterilization, and probably know that 50% of all medical devices that need sterilization go through radiation sterilization. And the goal of radiation sterilization is to basically get at least the minimum dose to make the product sterile. And at the same time, we don't want to overdo it. We don't want to overdose the product, because that can damage the integrity. So we define a minimum and maximum dose where we need to stay in. And we also can express that as the dose uniformity ratio, where we essentially divide the maximum dose by the minimum dose. And we want to stay below this defined level by this consideration. And a typical way without virtual dose mapping to do things is that you go through the device and packaging design, you go into small batch manufacturing, and then you go to sterilization validation. And the sterilization validation is done at the contract sterilizer. They basically take the product, put lots of dosimeters on there, run it through the process, and see what dose uniformity ratio you get. And if the dose uniformity ratio is within the boundaries you define, well, you're good to go and everything is good. However, that does not necessarily happen. So there can be outcomes where the dose uniformity ratio is too high. Then you go through a trial and error process to find what the reason is. Hopefully it can be resolved at the contract sterilizer. If not, I mean, you basically have to go and make changes to your design, even packaging, or worst comes to worst, basically even on the device. And then you go back through small batch manufacturing, go through the validation process again, and once you pass it, you go into production. With virtual dose mapping, this process can be condensed and done earlier. You can essentially do it as part of the device and packaging design. You essentially do the same thing that you do at this sterilizer. You basically take your device, you define where you want to put your dosimeters, and then you run it through the process. And the difference is that we do it virtually. We do it on the computer rather than going through the process and manufacturing everything. And now we can go through the loop. We can optimize our design. And once we are happy with that, we basically still go to small batch manufacturing and into sterilization validation. And we do that because sterilization validation is a regulatory requirement, which we can now do with confidence. We do essentially, we know where to put our dosimeters. We run it through the process, and we know that we will pass because our simulations told us the results already, and we go into production. So how do we know that these simulations, this modeling is actually giving us reliable results? I mean, the first thing we know that Monte Carlo simulations have been done many different, over decades and in many different fields, and they have shown to be very accurate. And for this sterilization field, we obviously do a lot of cross checks and experiments like the one that I'm introducing here. We took essentially an array of 3x3 syringes. We mounted it on a 3D printed plate to make sure that there's no movement in the packaging. We then we applied, like on each syringe, 5 dosimeters, and then to a contract sterilizer, Nutec Bravo, and did the measurements. And we see that here on the left, we see our 3x3 array of syringes from the top, and the beam came from the sides. And then we compare the measured and simulated dose for each device, as shown on the right, as for the 9 devices. And you overall see that there's excellent agreement between simulation and the measurement. I now want to walk you through a simple medical device here and show you the power of virtual dose mapping. And we start with this 2 syringes and 4 adapters that you want to package and see if it sterilizes. I mean, you obviously know that there are lots of choices packaging this device. I mean, how you orient the syringes, where the adapters are positioned, and so on. And this can all be addressed with virtual dose mapping, what the optimal configuration here is. But I won't go into this in too much detail. I mainly want to study how many of these boxes of devices I can sterilize in a row to maximize my throughput. And let's get started here. Again, like in the regular dose mapping, we define first all our dosimeters that we put on the surface of the devices. We define our beam directions, and then we go into virtual dose mapping. And here you see the results of the run with these dosimeters, and we color coded the dosimeters to show what the dose is. Blue is a low dose, and red is a high dose. So you see a distribution. But the beauty of virtual dose mapping is that you also get a 3D map on what is going on in your device. So we plot here the surface dose, and we see how the dose is actually changing over the device, and it's much more intuitive than just looking at these individual dosimeters. So here, we obviously can go to quantitative analysis by reading out the quantitative numbers of the dosimeters, and this is here shown for the device 1 and 2, and we get an overall the dose uniformity ratio of 1.21, which is pretty good. But now we want to see how many of these boxes can we really irradiate before our dose uniformity ratio goes bad. And we tried it here for stacking 3 boxes. Beam came from top and bottom, and we find that the total dose uniformity ratio comes to 1.6. And well, that's still a very good dose uniformity ratio. Let's try to go to 5 boxes. And at 5 boxes, well, it degrades. I mean, 3.12 as a dose uniformity ratio is probably not acceptable. And so I mean, likely you want to try 4 boxes, so you can further iterate to really find your optimum where you want to go with. I want to go to, yeah, right, that's a comparison for the 3 boxes. And I wanted to go to really see, is my packaging really adequate? I mean, I just told you that I put my devices in this box, and they're nicely ordered, and now I drive it to the contract sterilizer, and they're likely not that nicely in the box anymore. And we have actually developed a simulation tool to simulate that process. And you see, during transportation, something like that would happen to my medical device. So rather than being nicely ordered, as we saw before, we have now quite a bit of disarray in the box. And let's see what it does for our virtual dose mapping if we take that into account. So this is our unordered or disordered medical device, and we again do virtual dose mapping, and we can compare that now to our ordered device, and we see, I mean, the dose is really spread out much more than before, and our dose uniformity ratio goes up from 1.21 to 1.38, so significantly higher. So, well, what can we do about it? I mean, it's probably pretty clear that it's not a smart idea to just throw your devices into a box and hope for the best. So you have some sort of constraint for them, but what exactly is the constraint you need? And I mean, one obvious idea is, well, we put it into a tray and can then constrain the movement. And let's see what happens with a tray design like that. I mean, again, we see that moving it around gets the things moving, but I mean, they're definitely more constrained than they were in the free-for-all case. So we can then capture where these devices end up and do our virtual dose mapping. And we see here our dose map on the right, and we can calculate our dose uniformity ratio. And it turns out that the dose uniformity ratio is 1.35. So it's not really much better than with a complete disordered set of 1.38. So that's a little bit disappointing. What can we do about that? let's look at what really maybe contributed to that. I think we see a couple of things happening. One thing is that one of the adapters actually jumped over the fence. So we have now 3 adapters on the left and only 1 adapter on the right. The other thing is that the yellow adapter really snuck in between the 2 syringes that lengthens the radiation path. So that's probably also not good. What we do about it is we build a packaging that basically prevents the jumping over the fence, but we also reorient the syringes such that now the adapters are trapped behind the plunger here. And we see that, and just visually we see that there's a lot less variation now. And we can again do our dose mapping, and on the left, the surface dose, on the right, the virtual dosimeter readings, and we can calculate our dose uniformity ratio, and we see that it went down significantly to 1.2. But that's as good as the actual ordered case. So that's great. But let's go a little bit further. So one thing that's pretty obvious is shaking is a random process. So you can't expect that you shake and you get always the same configuration. So rather than just simulating one configuration, you have to simulate multiple configurations. And here I show snapshots of different possible configurations. We just run the same simulation multiple times and get essentially these snapshots. And we typically go to something like 8 to 10 different variations. And I show you the results here for 8 variations with the case without the tray. And we see that the range is really widely vary depending on the configuration. So if we had, for example, just used configuration 5 and said, well, that's great, we just have a very tight DOR, we would have missed the complete picture, because it's very clear that like configuration 6 or configuration 1 has a much wider spread. But all over, we have to consider all configurations. So basically the real range is between configuration 6 and 8. That gives you the full dose range. And we can do the same for the configuration with a tray. And we see that it is much more confined than without the tray. But we also find that there are some outliers. So there are some cases where the dose range is actually significantly higher. So there is, you see immediately the value of doing more than one configuration, because you need to address these outliers. And that would be now the next step in the packaging design to see how we can further constrain our device to make sure that we don't have these outliers. Well, with that, I want to conclude. We see that virtual dose mapping really correctly reproduces the measured data, and that really gives us the possibility to use design for sterilization in the design process. And we showed that design and device and packaging can be optimized using these modeling tools. And in particular, we showed that simulating shaking during transport is really important to get the full picture on what is happening in the process. With that, I want to close. Thank you, everybody, for your attention. And we stop share.
Thank you all very much to our presenters. We just have 4 minutes left, so we have a number of questions that have come in. One is, Should extractable and leachable testing be performed at max dose or closer to the routine processing dose?
Yeah, I can answer that. Great question. I would suggest that it should be performed closer to what you're actually going to use for your final product, so the routine dose. You don't want to do something that's not really applicable to what you're going to use. A higher dose, you would definitely see more extractables, but you'd just be doing that, kind of, it's not realistic if you're not going to actually use it for your product. So I'd say stick to the regular dose.
Okay, thank you. Another question is about biocompatibility. There's been a recent update to ISO 10993. Can you give us any insight in that, how that may impact the testing that's required for packaging with regards to biocompatibility?
I have not read that standard yet. We've got 7 weeks until it's published, so I was going to tackle that one here next. I apologize, I'm not ready for that question.
Okay. Tobias, for Canyon Labs again, for packaging stability studies, ISO 11607 guidance is that real-time aging must be started prior to the commercialization. You mentioned that you had seen real-time data fail accelerated aging. Would it be acceptable to restart real-time studies and keep product on market based on accelerated evidence?
So the answer to this question is that the failure actually was related to the way the company sealed the packages. And it was actually very not fortuitous, but lucky for them, because the sealing process was specific to the validation samples only, and we were able to identify it, and we did additional testing to demonstrate that the product in the market was okay.
Okay, thank you, Wendy. That's all we have time for today. A special thank you to our presenters for taking the time with us and providing their insights into today's topic. And thank you to everyone for watching this particular webinar today. The recording of this webinar will be circulated shortly, as will follow up to any question and answer session or questions that were not answered. Contact information for myself and our presenters will shortly be shown on the screen, and the webinar will officially end in a few minutes. Thank you again for joining us, and we look forward to seeing you at our next webinar.